Hey everyone, welcome back. In this video, we're going to take the results from our NF-core rnaseq pipeline on Vultr and transfer them back to our personal computer. Let's get started. We ran the NF-core rnaseq pipeline in a screen session, so let's reconnect to that and check on our results. Great, it looks like the pipeline completed successfully. It took about 3 hours and 41 minutes to process 4 paired-end RNA-seq samples, and all pipeline tasks completed successfully. So let's take a look at some of the results. The output files are stored in the run1 directory, and most of the files we'll be interested in are stored in the *_salmon directory, which includes the BAM files and the merged count matrix. And if you're not already familiar, the count matrix contains samples as columns, genes as rows, and read counts in each cell. Now let's get our results ready to send back to our personal hard drive. The first thing I'm going to do is create a tar archive, which will bundle all of our results into a single file that we can transfer all in one go. And this command creates a tar archive containing the entire run1 directory. The resulting tar archive is about 1.7 gigabytes, so it might take a few moments to transfer. Now I'm going to open another terminal pointed at my personal hard drive. And similar to how we got data onto the server, we're going to use the rsync command to download the tar archive from the server to our current working directory on my hard drive. Once the transfer is complete, we can extract the contents of the archive using the tar-xf command, followed by the name of the archive. Once the data is extracted, we can browse the contents of the output like we normally would. So in addition to the outputs we reviewed earlier, we also get a nice workflow report that includes the exact command we used, if we want to report this in our manuscript or rerun the pipeline at another date, how long the pipeline took to complete, and resource usage statistics for each pipeline process. We also get a nice quality control report summarizing information such as the number of reads that map to the reference genome, the percentage of reads that mapped, and other quality control statistics. So at this point, the bioinformatics is complete and we have everything we need to move forward with data analysis. In the next video, we'll look at the cost of running this pipeline in the cloud and how to shut the server down so we don't continue getting charged. I'll see you there.